Hey Avon, this is your biology department telling you to have a happy e-learning day. Do your best to message us with any questions that you have over Schoology, and please, please, please get your work done. We know it's not easy being trapped inside when you're not feeling very well, but do your best to get through everything, and we are here for you.
And wash your hands.
Please, please, like 20 seconds. The chorus of Oops, I Did It Again is sufficient, okay?
Or happy birthday twice.
Or a happy birthday twice, you know. There's options.
Whatever you're feeling, doesn't really matter. Just wash them.
Please. Lots of soap, lots of water.
And watch the videos.
I'm going to go over dihybrid crosses today over Brooke, Derek, Christian, and in order to figure out what's going on with Axel. Once again, if you have any questions, feel free to message us. So, diving in, the genetics of multiple genes. We have this law of independent assortment, which means that your chromosomes are going to all get mixed up during meiosis, and then when the egg is fertilized by the sperm, it is completely random which genes you get from your mom and your dad. So going through, you can see how we have all of the blue chromosomes in these eggs, all of the red chromosomes in these eggs, and then a combination down here. Now remember that an egg and a sperm has 23 chromosomes each, and so it is a total randomized process as to which 23 chromosomes you get in each egg and each sperm. This picture does a really great job of showing you how this moves through. We start out with an orange, white, gray, and black X. And you can see that in the end, there's a lot of variation between which eggs get which genes. This is represented best by a dihybrid cross, which is big and scary and complicated. But at the end of the day, we still have the dominant and recessive alleles that we worked with in the past. So the inheritance pattern is often measured by like color, genes, with the P's roundness or wrinkled. All of these traits are not blended together, but we are looking at things separately. So I can look here at color and then combine whether it is round or wrinkled. And so independent assortment is all about the combination of those multiple genes. In humans, this is going to look like hair color versus eye color. And the way we do this is by foiling. Same exact thing that you do in algebra when it comes to working with parentheses. So we're going to have the two genes here, and that's what's important. We always have two genes represented by the two letters. We have A's and we have B's. So in order to get the different combinations, you're going to do first, outside, inside, last, just like you do in math class. And in the end, you have the different A and B combinations. And this is what you're going to put at the top of the dihybrid cross. So that is what's most important. If you get anything out of this lecture, you are going to foil the genes and put those on top. You should have one of each letter on each top. So the Punnett square, looking at the genotypes, we have heterozygous A and heterozygous B. So what will the parent with heterozygous A and heterozygous B look like? In this case, we're saying that brown hair is dominant to blonde and that brown eyes is dominant to blue. Once again, things are a little bit more complicated, but this is just for a simple example. So we foil first, outside, inside, last, all the way across, and then when you draw those letters down, you're going to combine the A's with the B's. So doing the same exact thing as we did earlier, but instead of having just 2 letters in each box, you're going to have 4 letters in each box. 2 letters for each gene. So we move all the way through, and in the end, we can look at all of those and say, because we have a capital A and a capital B, we have a dominant A and a dominant B, that person is going to have brown hair with brown eyes. In the dominant A with the lowercase b, that is going to be brown eyes with blonde hair. In the recessive, homozygous recessive A's with the heterozygous or homozygous dominant B's, that person's going to have blue eyes with brown hair. And finally, there's a 1/16th chance that a person is going to have blonde hair and blue eyes. So, based off of this, we're going to say that Axel is homozygous recessive across the board. He has blonde hair, which means lowercase b, and blue eyes, lowercase a, lowercase a. So, you pick I want you guys to try to go through, feel free to pause the video, and do this with Derek, do this with Christian, and Brooke has blonde hair, blue eyes, so she is going to be homozygous recessive for the A alleles and for the B alleles. Whereas Derek, he's probably going to be heterozygous for both. And Christian, I know this is kind of hard to see with the video there, he's going to be homozygous recessive for all of them as well. Okay, hopefully you've gone through and done one of those practice problems. Here is the dihybrid cross with Brooke and with Derek. Now, Derek has brown hair, brown eyes, so we're going to say that he is heterozygous. And once again, like, he could be homozygous dominant, but we're just choosing to be heterozygous here to make more possibilities. And Brooke is blonde hair, blue eyes, and so she is going to be homozygous recessive across the board. So we do this cross. We foiled the letters again. Remember, we always foil those letters. And it looks like there is a 25% chance that Axel will have blonde hair, blue eyes based off of a dihybrid cross between Derek and Brooke. Now, how did I get 25%? We got 16 boxes here. 4 boxes out of the 16, that's 1/4. So 25% blonde hair, blue eye kid. Now, between Brooke and Derek, Brooke and Christian, that should say Christian up at the top. Christian is blonde hair, blue eyes as well, so they are all homozygous recessive. So the odds of having a blonde hair, blue-eyed child is 100%. 100%. So there is an easier way. I'm just going to show this to you in case if you do decide to go into AP biology later on. You can do these crosses separately. So we've got the hair color over here, and then we have the eye color in the bottom right-hand corner. And then in the end, you multiply those frequencies together in order to get your percentage. So once again, in the case of Derek and Brooke having a blue-eyed, blonde-haired child, it's going to be a 1/4 chance. So the last thing I want to talk about is a polygenic trait. This is what we typically see in humans. We don't just have brown eyes, blue eyes. We have a lot of different shades, tints of eye color. This is because there is more than one gene that goes into what your eyes looks like. So we have to use multiple alleles in order to figure out what that's actually going to be. Most people are going to fall into the spectrum of having some type of brown eye. There is a very wide range, and there's actually 8 different genes, which means that you would have a massive Punnett square. This is only for 2 different genes, A's and B's. So imagine having 8 different letters in each one of these boxes. So for the eye color, each one of these letters represents a gene, and then we're able to see the phenotype based off of that combination. So we have different parents that can come together, and this is what ultimately leads to the diversity of eye color, skin color, hair color, all of this. So once again, how do we get those letters that are up at the top of this dihybrid cross? We foil. First, outside, inside, last. So I've taken the parentheses and I've put those around the different letters. So parentheses around the A's, parentheses around the B's. First is the capital A, capital B. Outside, capital A, lowercase b. Inside, a lowercase a, uppercase b. And then last is the 2 lowercase letters. So we do this both times. And that's how we end up getting our combination. This is what you will have to do for your SpongeBob practice, or I'm sorry, your Incredibles practice worksheet. Now we do have some multifactorial traits. Your genetics determine a lot about who you are and what that looks like. But also how you live your life is also going to determine this. I've got a picture of Jessica Alba in the summer versus in the winter here. She has a gorgeous summer glow, but in the winter, when she's tucked inside, she's going to get a lot paler. So this is an example of how your genes do determine the original set point, but how you live your life is going to change your ultimate phenotype. So we have this very big scale between what your genes are and how your genes determine your life versus your environment. Down here on this x-axis, we're showing causes of death. So we have hemophilia versus cystic fibrosis, all the way over to a motor vehicle accident. Your genes have nothing to do with whether you're in a car accident or not. But all of these other things do have some contributions within genes, hemophilia and cystic fibrosis being purely genetic causes. So here's your summary. What is independent assortment? How does the law of independent assortment differ from the law of segregation? If you recall, the law of segregation is how your alleles and genes are split during meiosis, whereas the law of independent assortment is how those genes ultimately get combined and how you are your own collection of those alleles. A polygenic trait, poly meaning many, genetic meaning genes, we're looking at how different genes can go into one trait, like eye color or hair color or skin color. And then a dihybrid cross with the heterozygous AB and then a homozygous dominant AB. Good luck, and once again, if you have any questions, feel free to email us.